Cartilage is an essential skeletal and supportive tissue in our body. The geometrical properties of cartilage elements must be fine-tuned during the growth because cartilage participates in shaping of body parts and provides indispensable structural support to the organism during development. After early cartilage forms from mesenchymal condensations, growth typically occurs in all dimensions. At the same time, the very late growth of rod-shaped cartilages in limbs or expansion of sheet-like cartilages in basal skull are achieved through a mechanism of endochondral ossification that includes oriented cell dynamics. Since growth plates or synchondroses in skull are oriented towards a specific direction, the expansion of a cartilage in other dimensions is not clear from the mechanistic point of view and requires further investigation. We hypothesized that accurate cartilage growth might require alternative cell dynamics that do not involve hypertrophy, ossification, or growth plates. The developing face provides a remarkable variety of cartilage geometries and sizes, and therefore may serve as a sophisticated model system to study the induction of complex cartilaginous structures. To assess changes in the complete 3D anatomy of the mouse face following cellular-level mechanistic studies, we used a variety of approaches including microcomputed tomography, genetic tracing with multicolor reporter mouse strains, multiple mutants, and mathematical modeling. In this paper, we report a novel principle of vertebrate cartilage growth that is based on introducing oriented clonal units of chondrocytes into pre-existing cartilage. The orientation and geometry of chondrocyte clones defines the overall macro shape of the entire structure. We were able to alter cartilage shape by experimentally manipulating clonal geometries. For instance, sheet-like cartilages expand due to intercalations of numerous transversal clonal columns of chondrocytes. This previously unanticipated mechanism of growth uncouples the lateral expansion of curved cartilaginous sheets from the control of cartilage thickness. The latter is rather defined by numbers of cell divisions within individual clonal columns. Interestingly, in rod-shaped cartilage structures, the development of clonal columns determines the well-defined diameter and resulting rod-like morphology. Using in silico modeling, we discovered that anisotropic proliferation based on multiple clonal integration might explain cartilage bending and groove formation at the macro scale. At the same time, continual addition of freshly induced chondrogenic condensations that adjoin the main structure explains how complex minor geometrical features are born. All these mechanisms act in highly orchestrated fashion and may define the spectrum of evolutionary adaptations of facial shape among animals. Also, the further understanding of cartilage development may explain how humans attain individual facial features that we use to communicate our identity. Taken together, we reveal a novel set of principles contributing to the accurate scaling and shaping of cartilage tissue during growth.